Got
Hey
it.
guys. Oh, sorry.
Okay. Sorry. Go ahead.
Hey guys. Thanks for coming tonight. Yeah. Again, sorry about postponing the review session. Hopefully that didn't um have any scheduling conflicts or anything like that. But yeah. So tonight we're just gonna be going over the first two lectures from week 7, which is just gonna be the types of cytoskeleton and then microtubules. And again, like if you guys have any questions at any point, feel free to stop us. Um, but yeah, just get started then.
Oh, all right.
Okay. So yeah, so for the cytoskeleton, basically you just need to know there's three types. And really the one we go most in depth into is going to be microtubules. Um, but just to like go over them all like really quick, there's actin, which is also known as microfilaments, there's intermediate filaments, and there's microtubules. And all three of these are just like polymers, so they're made up of smaller subunits. Um, I don't think she mentioned the subunit for intermediate filaments, but for microfilaments, it's just going to be actin. And then for microtubules, it's going to be the tubule. But yeah, so for what each cytoskeleton does, if we start off with actin, that's what you're going to see in mainly muscle contraction, along with myosin. So like, oh yeah, so again, actin's mainly you're going to see that in muscle contraction with myosin, right? So actin's going to form the track that the myosin, like motor protein, like literally like walks along. And we'll go into that more later in this section or in the chapter. And then you're also going to see it in cytokinesis and vesicle and organelle movement. And it's also going to help maintain the cell shape. And that's like the a thing with a lot of cytoskeleton. It's really just mainly for structural support. And yeah, so moving on, uh, for intermediate filaments,
Oh,
those, oh, sorry, uh,
sorry.
no, sorry. For intermediate filaments, those are going to be also going to be maintaining cell shape. They're going to help with a lot of tension that the cell's going to be under. And the thing for them is that they're going to be anchoring the organelles and nucleus in place so that they're not just like freely floating throughout the cytoplasm. They're going to be um, in a certain place in the cell. And also another important thing about intermediate filaments is that they help make up the nuclear envelope. So, right, the nuclear envelope is going to have two layers, and the intermediate filaments are going to be in between the two. Then moving on to microtubules, these are the ones that like I feel like we go into most depth. They're going to be helping the cell resist compression. They're going to be moving along the vesicles. They're also going to be an important thing that we go over is during anaphase of the cell cycle. Those are what's going to be pulling apart the chromosomes to the opposite cells, right? And we're going to look at and those are actually like that's something she's going to ask a bunch of questions about, especially when it comes to like, oh, if you have like a cancer cell, what can you do to the microtubule to like what kind of drug would you introduce to help stop the cancer from like replicating, right? So how could you maybe destabilize the microtubule so it couldn't pull the chromosome to each end, right? Because if you can't do that, then the cell can't divide. And then the only other thing with microtubules is that they're going to be structural elements for things like cilia, which you find in like the linings of the gut, and what you find in like inside the lungs. And they're going to be used for flagella by like smaller cells, right? So they're going to be used for movement, and then they're also going to be found in centrioles. Um, so yeah, I think that's all I have for. Oh, and one last thing, microtubules and actin are both considered dynamic polymers, right? So they can grow and shrink as the cell needs. So, say you need something to be used as a track. So, like, again, like muscle contraction, right? You're going to need to have actin form the track that the myosin is going to walk around. In that case, you're going to want it to be a stable polymer, right? So you're not going to have dynamic instability there. But for something else, say like moving the chromosomes along, that's when you're going to want the cytoskel the microtubule to be dynamically instable, right? So it's going to be growing and shrinking, and so that's something that the cell highly regulates to do what it needs to. But yeah, I think that's that for cytoskeleton. Do you guys have any questions on any of that, or I feel like I went through that pretty fast, but all right, if that's the case, we can just go to the next slide. And then the other thing we're going to cover is fluorescence microscopy, right? So this is a very like common technique used to just see like what how like the structure is formed in the cell and how like it does its function. Um, so basically, like a good like rule of thumb is that when you're looking at like a slide like this or like a picture like this, the blue staining, the Daphne blue, is going to be for things like DNA or nucleus, ER, or like any other like organelles like that. The green fluorescent protein or GFP is going to be for microtubules. And then the red staining is going to be for actin. I'm not sure what type the red stain is. I know that in the video she said that GFP comes from jellyfish, which they can also like turn into different colors. So maybe it's that, but I'm not sure. Um, but yeah, so for these two pictures, you can see how on the diagram on the right, you can see how the actin filaments like surround the cell, and then the microtubules are what going to be are the cytoskeleton that's going to be growing on the inside. And you can see that on the picture on the left, right? The nucleus in this picture is going to be blue with the all the microtubules surrounding it. And then just like in the picture you see on the right, you're going to have the actin surrounding it. So it's kind of cool to see, to really see how the makeup is inside the cell. But yeah, so if we can go to the next slide. Here's just another picture that she has. It's another good side-by-side comparison that another thing that we can use um, fluorescent microscopy is to see the structure, because like these cytoskeletons are like so structure based. It's good to see that, and here you can see that in this case the ER is going to be colored in blue, and the green is again going to be microtubules, and you can see that you can really see how it like adds structure to it because it really follows along the same path that the ER is going to be throughout the cell, and you can see a better like simplified version of that on the left. And oh yeah, and before I forget again, another like technique she mentioned I think is fixing and staining, where you like freeze a cell and then you take it out and you basically so yeah, you freeze the cell and then you'll introduce like antibodies that are tagged with the fluorescent coloring, and then they'll attack like the protein, or they'll attach to the proteins the antibodies will in the cell, so that way you can kind of map out and see what kind of function the cell, protein's doing. So that's just like another technique she mentioned in the video. Um, but yeah, so if you can go to the next slide, I think we have uh, another. Oh yeah, and so this is another thing that she'll be going over is like the spindle assembly. So you're gonna see like a few different types of microtubules, and how they separate chromosomes. I know it says M phase, right? Oh yeah, I guess M phases, but during anaphase of M phase. Um, so yeah, basically, I think for now, we just need to focus on the kinetic core microtubules the most. We'll go into like the other two later on, but for now, you just need to know that well, aster microtubules are going to grow from the centrosome and attach to the cell cortex, and interpolar microtubules are going to also grow from the centrosome, and they're going to overlap at the spindles, and those, along with the kinetic cores, can grow and shrink. So they're going to have that dynamic instability that we talked about. And basically, all you need to know for that is that or for this is that the kinetic core microtubules are going to grow out or emanate from the spindle poles, and they're going to attach to the chromosomes at the kinetic core, which is where then they're going to pull back, or where the kinetic cores are going to like walk along the microtubules, using them as a track. And so they're going to like walk around and like pull their chromatid to their respective side. And that's basically how the cell can like replicate its DNA and get ready to divide. Um, but yeah, I think that's enough, or that's all for that slide. And yeah, here's just another picture of that. Of course, for this, you're not going to see the individual types of microtubules. It's all going to be in green, but you can see the blue chromosomes in the middle, and you can see the red is where they attach at the kinetic core. And you'll see, like, if I'm pretty sure she has videos on the Moodle page that are really good to watch for this, where you can see them like pulling the chromosomes apart, and you can really get a good idea for how they're like structured in the cell and how they function. Um, oh, and if sorry, if we can go back a slide really quick.
Yeah.
I feel like this diagram is something that you really want to. There's not much to understand, but you should know how to label the different microtubules and stuff. Because I do remember, I believe when I took the class, that she asked about this a lot in the exams. So that's just a little side note. But yeah, we can go back to the next slide. And the one after that. And yeah,
That's me.
so if you guys have, again, like any questions, and if not, then we can go on to microtubules.
All right, guys. So we're going to talk about microtubules. So first, we're going to talk about microtubule structure. So microtubules are obviously made of tubulin. So they're heterodimers. So they're alpha and beta tubulin. So they're both polar molecule, or they're polar molecules. So they have both a positive end and a negative end. So the tubulin addition can happen at either end, but it's faster at the positive end, which is why they call it the positive end. So they're kind of just like these long like tubes. You can kind of think of them as like straws. And they each have like, it starts with like protein filament or protofilaments, and the protofilaments go like around in a circle. And that's what makes the like straw structure. So the microtubules originate at the centrosome, and the negative end is embedded in the microtubule organizing center, and the positive end extends outward. So you can kind of see like here they have the beta and the alpha. Oh, it has a pointer. It has the beta and the alpha, and these are each like a subunit. And it has the plus end and the minus end with all the protofilaments, and it'll have 13 going down and 13 going around. And in between, like in the middle, is the lumen. So now we're going to talk about nucleation. So basically, it's the gamma tubulin ring is formed, and it acts as a base for like the start of microtubule growth, like kind of like the microtubule organizing center, similar to it. So it occurs in two steps. So the gamma tubulin like small complex assembly, and then the gamma tubulin ring complex. So the negative end is embedded in the gamma tubulin ring. So here you have a picture of the microtubules, and they're growing out of the tubulin rings, which are like the little red circles, if you can see them. I don't know. They're kind of small. And then here is the beta and alpha tubulin, like stacking onto the gamma tubulin to form like the 13 circle like protofilament. So now we're going to talk about dynamic instability. So the main idea of dynamic instability is that microtubules can grow and shrink independently of one another. So they're growing and shrinking all the time, and it's depending on the rate of GTP hydrolysis versus the rate of tubulin addition. So GTP hydrolysis, if it's greater than tubulin addition, then there would be shrinkage and depolymerization. So it would be unstable, and there would be no GTP cap. So since it would be shrinking and depolymerizing, the cap wouldn't be able to form fast enough, so it just wouldn't be there. And then if the GTP hydrolysis is less than tubulin addition, then there would be growth and the polymerization, and the GTP cap would be able to form and it would be more stable. So here there's just like a picture showing like the first one is a growing microtubule. So you can see the GTP tubulin molecules are adding, and since they're adding, it's forming the cap. And then in B, you can see the protofilaments that have the GDP tubulin wheel. So they're depolymerizing, and they're depolymerizing and falling away since they're bound to GDP, since it's cleaving off that last phosphate, and it's shrinking. So some of the different drugs that affect microtubules are, one is Taxol. So Taxol causes no depolymerization, so it's used as chemotherapy for cancer patients, because if there's no polymerization, then the microtubules can't form, and it prevents the cells from coming apart when they're going into anaphase for cell division. And then there's colchicine. And colchicine, I think she asked about it on the problem set, actually. I think she talked about it this week. But microtubule is a polymerization inhibitor, so it'd be used to control inflammatory responses. So for example, like gout. And then vinblastine, I don't know if she talked about vinblastine, but it's also a microtubule polymerization inhibitor, and it's used in another type of chemotherapy, and it prevents cell division. So now we're going to talk about MAPs. So these are microtubule associated proteins. So we're going to talk about tau. So tau is highly expressed in the nervous system and in neurons, specifically axons. And they bind microtubules and stabilize them because it's easy to purify because they're very heat stable. So as you know, like heat causes proteins to depolymerize. So since it's very heat resistant, it won't be able to break down. Sorry, I thought I was gonna sneeze and it just never came out. So dysfunctional tau may cause dementia or Alzheimer's disease. And if it's hyperphosphorylated, it can no longer bind the microtubules, and it aggregates with itself and causes like large tangles. So here you can see like a healthy neuron and tau binding to the microtubules on like the little orange parts. And then down here, you can see that there's a loss of tau, so it's easier for these microtubules to like depolymerize and come apart. Now we're gonna talk about motors. So motors hydrolyze ATP to move along the microtubule, and they transport cargo in the cell via the microtubules, and they have directionality. So kinesin walks towards the plus end, except for kinesin 14, which walks towards the minus end. And she usually asks about that, like on the exam. So that's something to remember. And kinesins are more diverse. Like there's a big family of kinesins, and they all like have different functions, and they all go different places in the cell. And then there's also dyneins, which is another type of microtubule transport, and it walks towards the minus end, and it's common in the flagella and the cilia. So these are also very heat stable. So here they have like an example. So going towards the minus end would be the dynein, and then going towards the plus end would be the kinesin, unless it's kinesin 14. And then here are some examples of different families of kinesins. So there's kinesin 5, which she's also going to talk about. And kinesin 5 is important because it's a tetramer. So it has 4 on each side, while the other ones they usually only have 2. And this one has 4. But it's because they overlap. So when they grab onto each other, they push each other in separate directions. So you see, like, the top is going in one direction and the bottom is going in another direction. It's because this kinesin is a tetramer. And then this is kinesin 1, which transports cargo, and kinesin 3, which moves the synaptic vesicles, and kinesin 13 destabilizes the ends of microtubules during spindle assembly. And an example of that is NCAC. And then I think that's the end. I think that's all we have.
Yep. So, do you guys have any questions about what we just went over, or like anything about like the material moving forward? Or...
So this is just covering the first two videos, right?
Yeah, yeah.
Okay, gotcha. I don't think I have any questions, honestly.
No, yeah, this stuff is like pretty introductory so
Yeah,
far.
it's just
So.
kind of getting to like the surface level of it all.
But yeah, it would be good to. Oh yeah, I can stop the recording. Yeah, it'd be good to remember the different types of kinesins, and then like the different types of like that diagram, microtubules, just stuff like that. I feel like she always includes in the exams, but
I feel like it's also very helpful to like draw everything. Like draw everything out that she talks about, because it helps to like really visualize it and like make it make sense in your head.
yeah, but